about described as mold identification and quantification, examining of biotic communities, including algae, zooplankton, and benthic invertebrates. We are detecting the invasive species in the water samples. We also work on asbestos in bulk and air samples. We perform different miscellaneous microscopy services, like identifying components of different types of dust and sediments, identifying some insects. Sometimes we're looking for the pieces of glass in the wine bottles. We also work on toxicity testing using the trout, Daphnia, and the bacteria in tests called microtox. Our other activities is measuring the radiant concentration in air. Today we'll focus only on mold, biotic communities, and invasive species. So what is it mold? It's a type of fungi. They are simple, often microscopic organisms which lack chlorophyll and are unable to produce their own food. So they have to grow on organic substances. They exist as thread-like filaments or hyphae and usually reproduce by spores. They found virtually everywhere, indoors and outdoors. Usually the outdoor concentration of mold spores is much higher than indoors during the spring, summer, and fall months, although it will depend on the type of ventilation in the buildings, on the activities in the buildings, and if any mold is growing there. If molds are growing in humid or water-damaged buildings, they are called biocontaminants. Large number of biocontaminants in buildings could be associated with health problems, which are mainly the upper respiratory, mainly upper respiratory symptoms, causing allergies, decreased general immunity, infections. They could also cause structural integrity problems for buildings, for example, the dry rot fungus. The identification performed in our lab is restricted to the common indoor molds. We cannot identify Blastomyces, Cryptococcus, or Histoplasma. The mold samples, we can analyze some air samples on the viable samples on rose Bengal agar strips. These are our code is mold-WP, or non-viable samples, spore traps with the code aerosol-WP. We can analyze also swab and bulk samples with the code mold-WP, which reports the types of mold and the number of colony-forming units per centimeter square or per gram of every type of molds, or mold ID-WP, which reports all types of molds found in the samples with description of small, moderate, large, or massive amount. The bulk samples and tape lifts can be also examined by the direct microscopic exam, and our code is MISEX-WP. If the speciation is required, it is possible only on viable samples. So for speciation, please add mold speciation-WP for total speciation or speciation of all molds in the sample, or mold spec-HC-WP for speciation of Aspergillus, Penicillium, Fusarium, and Stachybotrys only. All viable mold analysis require at least 7 days of incubation, and the speciation requires another 14 days, so 21 days of incubation for mold speciation is required. All viable samples should be kept cold but not frozen and delivered to the lab as soon as possible or within 3 days. The viable air samples are taken with the RCS sampler and rose Bengal agar strips. Those strips are inserted into the sampler, and when the air is blown onto them with all the spores and hyphal fragments impacting on the agar. Very important thing is to make sure to put the agar strips into the plastic envelopes exactly the way as they were taken out after the sampling is completed. This ensures that the molds have enough room to grow and sporulate. You can see on the picture, you can see the flat surface of agar on the agar strips, which should be facing the molded side of the plastic envelope. If the strips are incorrectly inserted, then the molds might not have enough room to grow and sporulate, and results could be biased. After 7 days of incubation, all molds on the strip are identified and counted and reported as number of colony-forming units per cubic meter of air for each type of molds. The non-viable air samples can be taken with the Ziphon sampler, which is drawing 15 liters per minute of air, and the aerosol cassette. On the market, there are different cassettes, different spore traps, which could be used, and the different types of spore traps have different recommended air volume. So please ensure that the airflow is appropriate for the type of cassettes used. Upon receiving the spore traps at the laboratory, the samples are stained and all the mold spores are identified, counted, and report as the number of fungal structures per cubic meter of air. For the air samples, please make sure to always provide the air volume sampled. Which kind of samples to use, viable or non-viable? It depends on the purpose of the study and on the time constraints. So to compare both types, the viable samples require at least 7 days of incubation. You cannot speed up this process. Non-viable samples could be analyzed on the day of receiving, so it's much faster analysis. Only viable molds can be seen and identified on viable samples, while on non-viable samples or spore traps, both viable and dead spores are identified and counted. The speciation is possible only on viable samples. Also, the Aspergillus and Penicillium could be identified to genus only on viable samples. In spore traps or non-viable, we are identifying them as Aspergillus/Penicillium group, unless entire conidiophores are seen, but this happens very rarely. The viable samples could be done in dusty environments, while the spore traps from the dusty environments will be obscured, will be covered with dust, and often not all spores can be seen and counted. So the results will be biased. On the other hand, the viable samples get very easily overgrown with fast-growing fungi, very easily get contaminated, while in non-viable samples, all spores can be seen and there is no possibility of contamination. But when fungi will not sporulate in viable samples, when fungi will not grow in culture at all, while on spore traps, all spores from different groups of fungi could be seen and identified. So there are pros and cons of both types of sampling. The swab samples are taken with sterile swabs, and please always make sure to provide the area sampled. If the area sampled is not provided, we assume it is 100 centimeters square, and we report results as number of colonies per centimeter square. The bulk samples, they could be analyzed both as viable or non-viable samples. We need a very small sample, about 1 gram, so if possible, please do not send us entire tiles. Both swab and bulk samples are mixed with sterile water. The serial dilutions are made and plated onto the agar plates. The agar plates are incubated for 7 days and 25 degrees Celsius. And after this incubation time, the colonies are identified and counted basing on colony morphology. On the upper picture, you could see the same mold. It's Aspergillus nidulans, grown on 3 different medias, and the colors and shapes of colonies will depend on the media used. After that, the samples are examined under the dissecting microscope, and at this point, the majority of colonies can be identified to genus. If there is a need, the slides are made and examined under a compound microscope, where we pay attention to the size, color, and shape of spores, type of sporulating structures, width, color, roughness of hyphae, and many other different characters which are specific for certain types of molds. The mold samples could be taken also with the tape lifts, which are used for non-viable surface sampling. We recommend using the biotape slides, which we can provide, but if clients don't have the biotape slides, the crystal clear Scotch tape could be used. Please use very small pieces of that, approximately 1 inch long. These tape lifts are stained and examined under a compound microscope at the laboratory. At this point, we can usually identify molds up to genus. We do not report the number of molds structure on the tape lifts. We only specify if the amount is small, moderate, large, or massive. We'll move now to the examining of biotic communities. This is a very important tool for respond to changes in habitat by adjustments in community structures. We are examining phytoplankton or algae, which are the microscopic plants suspended in water column, zooplankton, which are microscopic animals suspended in water column, unable to swim against the current, and benthic invertebrates. The phytoplankton and zooplankton samples are settled in 5, 10, or 25 milliliters containers with very thin glass bottom, which enables us to examine and identify and count all the organisms which are settled on the bottom. After a few hours of settling, the samples are examined using the inverted microscope. We have different analytes for different types of analysis for phytoplankton samples. We require approximately 100 milliliter of sample in any container. The sample should be preserved with Lugol's preservative or kept cool. We run different types of tests. Algae ID-WP gives the identification of dominant types of algae and the amount specified as small, moderate, large, or massive. The Phyto-WP, it's much longer analysis. It reports the identification and count of all algae in the sample up to genus and species if possible. And Phyto-Bio-WP, it reports identification and count of all algae in sample with average biovolume measurements. We have special analyte for the Cyanobacteria or blue-green algae. Why is that important? It's because this type of algae, which are Cyanobacteria, mostly Anabina, Microcystin, and Aphanizomenon, often produce toxic blooms. They could release toxins, and World Health Organization specifies a limit of 100,000 cells per milliliter as a high-risk health alert for recreational water, which corresponds to approximately 20 parts per billion of microcystin. These analyses give us identification and cell count of all blue-green algae found in a sample. The zooplankton, for zooplankton we require approximately 100 milliliters sample. They should be preserved in 70% alcohol or kept cool. If the samples are not preserved, please inform the lab, so we can preserve it here right away. We offer two types of tests. Would be ZOOP-WP, which reports identification and count of all zooplanktons in the sample, and ZOOP-Bio-WP, which reports identification, count, and biovolume measurements of all zooplanktons in the sample. We also identify the benthic invertebrates. Those are the bottom-dwelling aquatic invertebrates, mainly insects, crustaceans, mollusks, worms, and mites. These organisms are sedentary and relatively long-lived, and many of them have different tolerances, different sensitivities to pollution, so they are excellent bioindicators. The examples of bioindicators could be found mainly in three orders of insects. These are nymphs of mayflies or Ephemeroptera, nymphs of stoneflies or Plecoptera, and larvae of caddisflies or Trichoptera. Particular families within these groups have different tolerances also, so those are excellent bioindicators of quality of our waters. The benthic invertebrates, which called Benthos-WP, report the count and identification of benthic macroinvertebrates. Samples should be preserved in 70% alcohol. They may require extensive sorting. For the samples with large amount of invertebrates, we can subsample them using the pie method or Marchand box method. Both Cabin and OBBN protocols are available, and primary analyst is certified by North American Biological Society for family-level identification. For all projects, we send the questionnaire to the clients, asking them to specify the condition of subsampling, if they need the reference library, if they need the organisms and sediment returned, as well as if they need the non-benthic groups identified and counted as well. We also ask for the required identification level if identification to family or to genus is required. So please always fill up the questionnaire before asking for the quote. We also work on the invasive species, mainly on zebra mussel, which we identify under the stereoscopic microscope fitted with 2 polarizing filters. We are looking mainly for the veligers of zebra mussel, which are very small, which cannot be seen with the naked eye, but under the polarized light they show very distinct Maltese cross, which helps with identification. We also identify the spiny water flea in water samples. Those 2 invasive species make a havoc in our waters, outcompeting the native species and creating huge problems. We are accredited by American Industrial Hygiene Association for mold and asbestos analysis, and we are also accredited by Canadian Association for Laboratory Accreditation for mold, phytoplankton, zooplankton, benthos, bioassays, and radon. And this would conclude my presentation. Do you have any questions? Thank you very much. Okay. Yes, the question is, can you explain a bit more about the biovolume measurement done in phyto and zooplankton test? Yes, we are taking the measurements of the length, width, and depth of the samples. We are basic of the organism. We are fitting them into the cubes or perpendicular prisms, and we are just getting a few measurements for every type of organisms and getting the average biovolume for all kinds of phyto and zooplanktons. Any other questions? Okay, the question is, in the case of airstrip for rose Bengal, what was the name of the instrument used? This is the RCS sampler, or rotary centrifugal sample, used with agar strips. This sampler takes 40 liters per minute, and usually the samples are taken for 4 or 8 minutes. Any other questions?